12, we'll be discussing about B cell activation and antibody production. So, in this chapter, we are going to look on overview of humoral immune response, antigen recognition and antigen-induced B cell activation, antigen capture and delivery to B cells, activation of B cells by antigen and other signal, helper T cells dependent antibody response to protein antigen, the sequence of events that will happen, and we'll further discuss also the antibody response to T independent antigen, subsequent B cells that respond to the independent antigens, and we'll discuss the antibody feedback regulations of humoral immune response, and there are many things in between that I've not read here, but we will be discussing together to see how everything works. So, the key concept here is B cell activation and antibody production. So, in general, we are discussing about humoral immune response. Here we go. Humoral immunity is mediated by secreted antibodies, which are produced by cells of the B lymphocyte lineage. This chapter describes the molecular and cellular events of humoral immune response, in particular the stimuli that induce B cells proliferation and differentiation, and how these stimuli influence the types of antibody that is produced. The mechanism by which antibody eliminate microbes are described in chapter 13. Overview of humoral immune response. The activation of B cell results in the proliferation and eventually differentiation into antibody-secreting plasma cell and memory cells. Humoral immune response are initiated by specific B cell recognition of antigen in secondary lymphoid organs. Antigen bind to membrane immunoglobulin M, IgM, and D on mature naive B cell, generating signal required for their proliferation and differentiation into plasma cell. The antibody that is secreted by plasma cell has essentially the same specificity as the original antibody that saved as antigen receptor on surface of the naive B cells. A single B cell may within a week give rise to more than 5,000 antibody secreting cells, each of which can secrete about 2,000 antibody molecule every second. This cell expansion and the remarkable rate of the antibody secretions are needed to keep pace with rapidly dividing microbes. Antibody response are T dependent or T independent depending on the nature of their antigen and involvement of the helper T cells. Most response to protein antigen require T cell help, so these antigens are called T dependent, and the term helper T lymphocyte comes from the realization that T cells stimulate or help B lymphocyte to produce antibodies. In T-dependent response, some activated B cells begin to produce antibodies other than IgM. This process is called heavy chain isotopes class switching. As the response develops, activated B cells produce antibodies that bind to an antigen with increased affinity, and these B cells progressively dominate the response. This process is called the affinity maturation. In addition to isotope switching and affinity maturation, helper T cells stimulate the production of long-lived plasma cells and generation of memory B cells. Multivariant antigen with repeating determinants such as polysaccharide can activate B cells without T cell help. These antigens are called T-independent. T-independent response are rapid but relatively simple, consisting of mostly of low affinity IgM antibodies, whereas the T-dependent response are slower to develop but the results is more durable, high affinity antibodies that are typically of IgG, IgA, IgE isotopes. Primary and secondary antibody response to protein antigen differ qualitatively and quantitatively. Primary response results from the activation of previously unstimulated naive B cells, where the secondary response are due to the stimulation of expanded clone of memory B cells. Therefore, The secondary response develops more rapidly than does the primary response, and larger amount of antibodies are produced in the secondary response. Furthermore, because the memory cells are already undergone isotope switching and some affinity maturation, there is more IgG and other isotope compared to IgM, and the affinity of the antibody is higher in secondary response. So we have diagram figure 12.1. The phases of humoral immune response. The activation of B cell is initiated by specific recognition of antigen by surface immunoglobulin, receptor of the cell. Antigen and other stimuli, including helper T cells, stimulate the proliferation and differentiation of the specific B cell clones. Progens of the clone may differentiate into plasma cells that produce IgM and other Ig isotopes, IgG for example, may undergo affinity maturation and may persist as memory cells that have also typically undergone class switching and affinity maturation. So we have seen this one, the antigen recognition, the naive IgM, IgD B cells, by the help of T helper stimulus, it can proliferate then to get plasma IgM, IgD cells, then they secrete other antibodies, or it can express IgG, it can express also high affinity expression of B cells, which they can go for high affinity Ig expressing B cell, they can go and form memory B cells. Some may mature and affinity maturation may happen. So distinct subset of B cells respond preferentially to different types of antigen. Follicular B cells in secondary peripheral lymphoid organs make mostly antibody response to protein antigens, and these B cells response require collaboration with helper T cells. Marginal zone B cell in the spleen and other lymphoid tissue in human, and B1 cell in mucosa tissue and the peritoneal recognize multivariant antigens such as blood-borne polysaccharides and mount primarily T-independent antibody response. These preferences are not obsolete. Some marginal B cells participate in T-dependent response, and some follicular B cells may make T-independent response. So here we have another figure, which is the T-dependent and T-independent antibody response. T-dependent antibody response to protein antigen mainly involve follicular B cells. T-independent response to multivariant antigen are mediated mainly by marginal zone B cells in spleen and B1 cell in mucosa sites, IgM immunoglobulin. So we have T-dependent protein antigens, follicular cells. Then with the T helper cell, this is dependent. You need the T helper cell. Then the isotype switching high affinity antibodies, memory B cell, long-lived plasma cells. T independence here, the polysaccharide comes attached to the B cell. Then B cell is activated. Then mainly IgM, low affinity antibody, short-lived plasma cells. So here we are not expecting a lot with the long-lived plasma cell and memory cells. There are very few because mostly IgM is maintained. Another figure here is figure that at 12.3, primary and secondary immune response. In primary immune response, naive B cells are stimulated by antigen, become activated and differentiate into antibody secreting cell that produce antibody specific for eliciting antigen. A secondary immune response is elicited when the same antigen stimulate memory B cells, leading to the production of great quantity and specific of antibody than produced in the primary response. Note that the characteristics of secondary antibody responses summarized in the table are typical of T-dependent antibody response to protein, antigen, and immunoglobulin. So you see them. In the primary response, we have mostly here the IgM. It is a naive T cell activated, short-lived plasma cell, lymphoid organs, long-lived plasma cell. They remain they go to the bone marrow. Then when you have the second infection of the similar, they go very faster, and there's more specific immunity. So features: the magnitude for primary response small, but for the secondary is high or larger. Antibody isotopes, usually IgM greater than IgG, but secondary is more of increased IgM and other, often IgA and isotopes of IgE. Antibody affinity, it's low. Average affinity more variable. It's here is very high high affinity maturation induced by all antigen, only protein antigen that have been already known. With this background, we proceed to discuss of the B cell activation, starting with the interaction of antigen with B cells. We will then describe the role of helper T cells in B cell response to protein antigen and mechanism of isotope switching and affinity maturation. We conclude with discussion of T-independent antibody response. Antigen recognition and antigen-induced B cell activation. To initiate antibody response, antigen have to be captured and transported to B cells area of the secondary lymphoid organs. These antigen then initiate the process of B cell activation, often working in consent with other signals that are generated during innate immune response, triggered by microbes or by just adjuvant in vaccine. We will next describe these early events in B cell activation. Antigen capture and deliver to B cells. Antigen may be delivered to naive B cells in lymphoid organ by multiple routes. Antigen that elicit the antibody response may vary in size and composition. They may be small, soluble, larger, or particulate, and may be free or bound to antibodies. The major pathway of antigen delivery for different types of antigen include the following. So there are major pathway that include from the diagram here, you will see the most antigen from tissue sites are transported to lymph nodes by afferent lymphatic vessel that drain into the subcapsular sinus of the nodes. Soluble antigen generally smaller than 7 kd may then reach the B cell zone through conduits that extend between the subcapsular sinus and underlying follicles. See chapter already saw in chapter 2. In some cases, small antigen may gain access to follicular by diffusion. Subscapular sinuses macrophage capture large microbes and the antigen antibody complexes and deliver them to follicles. Antigen in immune complexes entering the spleen may bind to complementary receptor, in particular complement receptor type 2, type complementary CR2 and CD21 on the marginal zone B, and these cells they can transfer the immune complex containing antigen to follicular B cells. Recirculating follicular B cells may also capture antigen via CR2 and deliver them into follicle. Immune complexes that are delivered into the lymphoid follicle may bind to CR2 and FC receptor for IgG on surface of follicular dendritic cells. And the antigen in those complexes are then presented to antigen-specific B cells in sustained manner over days to weeks. Polysaccharide antigen can be captured by macrophage in the marginal zones of splenic lymphoid follicles and displayed or transferred to the B cell in this area. In all of these cases, the antigen that is presented to B cell is generally in its intact native formation, and it's not processed by antigen presenting cell. This, of course, is one of the important difference between the form of antigen recognized by B cells and T lymphocytes, as discussed in chapter 6. Although the presentation of antigen to B cell by subscapular sinus macrophages and macrophagy in the splenic marginal zones and dendritic cell in medulla lymph nodes has been described in experimental model, how these cells prevent the protein antigen they capture from being engulfed and degraded remain unclear. So before we go further, let us see the diagram here. The pathway for antigen delivery to the follicular B cells. Small antigen are delivered to B cells in follicles through afferent lymphatic and via conduit of large antigen by subscapular sinuses, macrophage, or by dendritic cell in medulla. In all these cases that we have described above, the antigen that is presented B cells generally is in its intact cell, we have said already, native conformation, and is not processed by antigen presenting cells. This, of course, is one of the important difference between the forms of antigen recognized by B cell, B and T lymphocytes. Although the presentation of antigen to B cells by subcapsular sinuses macrophage in the splenic marginal zones and dendritic cell in medulla of lymph nodes has been described in experimental model, how this cell prevents the protein antigen they capture from being engulfed and degraded remain unclear. So activation of B cell by antigen and other signals. B cells receptor complex of mature B cell is composed of membrane Ig molecules that bind antigen and associated Ig alpha and beta Ig beta protein that deliver signal for B cell activation. B cell receptor complex play two key roles in B cell response. First, binding of the antigen to the receptor delivers biochemical signal to the B cell and initiate the process of activation. As discussed later, signaling is more robust with the multivariant T-independent antigen that within T-dependent protein antigen than with T-dependent antigen. The antigen-induced biochemical signal are initiated by surface receptors, family kinase, mediated phosphorylation, and immunoreceptor tyrosine-based activation motifs, tyrosine of Ig alpha and Ig beta, followed by recruitment of activation of cell, shown already in chapter 7. Second, the B cell receptor internalize the bound antigen into endosomal vesicles, and if the antigen is a protein, it is processed into the peptide that may be presented by plus 2 MH2 molecule on the B cell surface for recognition by helper T cell. This antigen presenting function of B cell will be considered later in the context of T-dependent B cell activation. Although antigens recognition can initiate B response, by itself it is usually inadequate to stimulate significant B cell proliferation and differentiation, even for T-independent antigen. For full response to be induced, other stimuli cooperate with the B cell receptor engagement, including complementary proteins, pattern recognition receptors, and in this case, protein antigen helper T cells, discussed later. So the role of complementary receptor type 2, we see this one, you see the diagram here, the role of complementary receptor type 2 and toll-like receptor in B cell activation. In immune response to microbes, activation of B cells through B cell receptors may enhance the may be enhanced by complement coated antigen that can regulate both B cell receptor and complement receptor 2, and also the simultaneous activation of toll-like receptors and B cell molecules. Pathogen associated molecule pattern also is important. So we see here, for proliferation, you have microbial antigen, then it bound with C3D. This is one of the complement kind of thing. Then when it's bound there, it is this one is mostly effective with the activation of other co-stimulators. Then this signaling will happen. There will be proliferation or differentiation, and in pump microbes can happen. Then toll-like receptor signaling can happen. Then activation will happen. B cell activation is facilitated by CR2 co-receptor on B cells, which recognize complementary fragments that are covalent attached to the antigen or part of immune complex containing antigen. A follicular B cells and marginal zone B cells expresses the complement receptor CR2. The level of CR2 on marginal zones B cells are much higher. Microbes activate the complementary system in the absence of antibodies by alternative or lectin pathway, and in the presence of antibodies by classical pathway. In all these situations, complement fragments are generated that bind to the microbes. One of the fragments called C3D is recognized by CR2, which enhance the strength of B cell receptor signaling and thus function as co-receptor for B cells. Some non-microbial polysaccharides also activate the complement by alternative or lectin pathway, and this is one reason that much antigen are able to induce antibody response without T helper. Microbial products engage T toll-like receptor (TLL) on B cell, which also enhances B cell activation. Human B cell expresses several toll-like receptors, including toll-like receptor 5, which recognize bacterial flagellum, endosomal toll-like receptor 7, which recognize single-stranded RNA, and toll-like receptor 9, which is specific for unmethylated CpG-rich DNA endosomes. Murine B cells, but not human B cell, also express a toll-like receptor on cell surface, which recognize lipopolysaccharide LPS. These pattern recognition receptors provide signal that enhance or cooperate with those from B cell receptor complex during B cell activation. In addition, the activation of myeloid cell through a pattern recognition receptor can promote B cell activation indirectly in two ways. A dendritic cell activated through the toll-like receptor contributes significantly to help T cell activation, and the helper cell stimulate B cell in response to protein antigen. Myeloid cell activated by toll-like receptor may secrete APRIL, a proliferation-induced ligands, proliferation-induced ligand, and BAF, B cell activating factor. Cytokine that can promote T-independent B cell response. So you have here the myeloid, the APRIL, which is the proliferation-inducing ligand, and BAF, which is B cell activating factor. They are very important and they are produced by myeloids, which together they function to facilitate the B cell proliferation through the process. Also, apart from that, we see the helper T cell. The interaction of different types of antigen, multivariant structural or protein with B cell initiate B cell proliferation and differentiation in different ways. Signal from B cell receptor may be sufficient to keep the B cell alive, induce changes in chemokines receptor expression, and promote antigen endocytosis. The importance of signaling by B cell receptor complex for subsequent response of the cell varies with the nature of antigen. Most T-independent antigens such as polysaccharide contain multiple identical epitopes on each molecule. Such a multivalent antigen can effectively cross-link many B-cell antigen receptor initiate response even though they are not recognized by helper T lymphocyte. In contrast, many naturally occurring globular protein antigen possess only one copy of each epitope per molecule. Therefore, such protein antigen is protein in their functional monovalent form cannot simultaneously bind to and cross-link multiple Ig molecule, and their ability to directly activate B cell is limited. They do not typically induce signal that can drive B cell proliferation and differentiation. Some protein antigen may be displayed as multivariant array on the surface of microbes or cell, and they may be multivariant because they are in aggregate, and in these cases, protein antigen can induce B cell proliferation and differentiation. So, of note here, we repeat again, very important, the interaction of different types of antigen, multivariant structures, or protein with B cell receptor initiate B cell proliferation and differentiation in different way. Signal from the B cell receptor may be sufficient to keep the B cell alive, induce changes in chemokine receptor expression, and promote antigen endocytosis. The important signaling for B cell receptor complex for subsequent response of cell varies with nature of the antigen. Most T-independent antigens, such as polysaccharides, contain multiple identical epitopes on each molecule. Such a multivalent antigen can effectively cross-link many B cells antigens receptor and initiate response even though they are not recognized by helper T lymphocyte. In contrast, many naturally occurring globular protein antigen possess only a copy of each epitope per molecule, Therefore, such protein antigen is their functionally monovalent forms cannot simultaneously bind to the and cross-link the multiple Ig molecule, and the ability to directly activate B cell is limited. They do not typically induce signal that drive B cell proliferation and differentiation. Some proteins antigen may be displayed as multivalent arrays on the surface of microbes or cells, and or they may be multivalent because they are in aggregates. In these cases, protein antigen can induce B cell proliferation and differentiation. So here below we have table 12.1, the effects of B cell antigen receptor engagement on B cells. So we have phenotypical changes, we have functional consequences. Increased expression of CCR7 lead to the migration toward T zones. This is important because that's who they meet. They help to meet each other. Increased expression of B7 costimulators enhances the ability to activate helper T cells, and the increased expression of receptor for T cell cytokines increases responsiveness to signal from T helper cells. Increased expression of anti-apoptotic protein, it increases the survival of B cells. These changes may be induced by binding of the protein antigen to B cell receptor, and they prepare B cells to respond to T cell helper. Protein antigen are also internalized, processed, and presented to helper T cell. After specific B cells recognize antigen, the subsequent step is humoral immune response are very different in T-dependent and T-independent response. We will next describe the activation of B cell by protein antigen and helper T cell. Helper T cells dependent antibody response to protein antigen. The helper function of T lymphocyte was discovered by experiment performed in late 1960s, which showed that antibody response required cooperation between B cells and T cells. These classic experimental studies were among the first to demonstrate the importance of interaction between two different cell populations in the immune system. It was later established that most helper T cells are CD4, CD8 negative, CD4 plus CD8 negative lymphocytes that recognize peptide antigen presented by class 2 major histocompatibility complex molecules. One of the important accomplishments of immunology has been the elucidation of the mechanism of T and B cell interaction and actions of helper T cell in antibody response. The sequence of events during T cell dependent antibody response. Protein antigen are independently recognized by specific B and T lymphocyte in secondary lymphoid organs, and the two activated cell types interact with each other to initiate humoral immune responses. Naive CD4 T cells are activated in T cell zones by antigen in form of processed MSC associated peptide presented by dendritic cell. Naive B cell in follicles activated by the same antigen in its native conformation that is transported there. The activated helper T cell and activated B cell migrate toward one another and interact at edges of follicles where the initial antibody response develop. Some of the activated T and B cells migrate into follicle to form germinal center, where more specialized antibody response are induced. Next, we will describe each of these steps in detail. Initial activation and migration of helper B cell and T cell. The contemporaneous activation of specific B and T cells by a protein antigen induces change that bring them into proximity to enhance likelihood of the antigen-specific B and T cell colocalizing and interaction with one another. The frequency of naive B cells or T cells specific for given epitope of an antigen is as low as 1 in 10 power 5 to 1 in 10 power 6 lymphocytes, and the specificity B and T cells have to find each other and physically interact to generate strong antibody response. This is accomplished in part by regulated the movement of cell following antigen recognition. Before antigen encounter, naive T cell reside in T cell zone because they express a chemokine receptor CCR7, which bind to the chemokine CCL19 and CCL21 produced in these zones. And naive B cell remain in the follicle because they expresses CXR5, which recognize CXL13 produced by follicular dendritic cells and other cells in follicles. Upon activation, helper T cell downregulate CCR7 and increase the expression of CXCR5, and as a result leave the T cell zones and migrate toward the follicles in response to CXL13 in follicles. B cell responds to antigen mediated B cell receptor triggering by reducing cell surface expression of chemokines receptor CXR5 and increasing expression of CCR7. These cells also upregulate the AB12, the chemoattractant receptor that responds to the oxysteroid ligand made along the follicle T zone interface. EB12 is so named because it was discovered as an Epstein-Barr virus-induced gene. Activated B cells thus migrate toward T zones drawn by a gradient of CCL21, the major ligands for CCR7 and oxrol. The net result of these changes is that antigen-activated T and B lymphocytes are drawn toward each other. Protein antigen are internalized by B cells and presented in form that can be recognized by helper T cells, and this represents a next step in the process of T-dependent B cell activation. Antigen presentation by B cells and Hampton carrier effect. So protein antigens that are recognized by specific B cell receptor are endocytosed and processed to generate a peptide that bind to class II MHC molecule and are presented to CD4 T helper cell. This class II MHC pathway of antigen presentation was described in detail in chapter 6. The peptides that are presented by B cells to helper T cell are the same peptide that initially activated the naive CD4 T cells when they were presented by dendritic cell in T cell zones. Because B cell receptor recognizes an epitope on the native protein with high affinity, specific B cells binds this antigen much more efficiently. Because the B cell receptor recognizes an epitope of the native proteins with high affinity, specific B cell binds this antigen much more efficiently. This is as much lower concentration that do other B cell not specific for antigen. Therefore, specific B cells are also much more efficient at presenting a peptide derived from that antigen than other B cells that do not express its membrane receptor for the antigen. This is why B cells specific for an antigen are best able to interact with helper T cell specific for that antigen and receive help signal, whereas B cell with other B cell receptors remain in quiescent state. So we have the diagrammatic presentation here, and I think let us go through them. And sequence of event in humoral immune response to T cell dependent protein antigen. A. Immune response are initiated by recognition of antigen by B cells and CD4 T cells. The activated lymphocytes migrate toward one another and interact at the interface of T and B cell zone. The initial T-independent B cell proliferation and differentiation result in formation of an extrafollicular focus, in which B cell proliferate and can undergo isotope switching and they differentiate into plasma cell, mostly short-lived. Some of the T cells that activated in extracellular focus develop into follicular helper T cells, migrate back to the follicles together with some activated B cells to form geminocenters. The later events in B cells response occur in geminocenters and include somatic mutation and the selection of high affinity cell affinity maturation. Addition to isotope switching, memory B cells generation and the generation of long-lived plasma cell described later in figure. So in here, in summary, we have A, this immune response initiated by recognition by antigen by B cell. We see we have the dendritic cell in other sides, then T cell epitope, the naive T cell activate, work together, it is activated already, initiate TB interaction. So this one, if it is activated, now it is, as we are told, that it loses some of the important expression like CCR7, then it is coming away. And also in other way, this protein which was expressed here, it is also expressed in a B cell. And in B cell, what happened, the B cell is activated and now it is also trying to present, and it is losing some of the characteristics that was making it to stay in primary follicle. Now it is moving toward here. So everyone, the other, this one is moving this way and this moving this way, and here they are meeting. So B cell that miss the helper cell which has already activated, they lead to the production of extrafollicular focus, release of these more antibodies, more B cells which are activated. Then they can go back and have to the geminocenter and work together and produce T follicular helper T cell here, and they can work together to produce more of a strong B cells, activate the B cells. So the figures 12.7, migration of B cell and helper T cell cells and T cell B cell interaction, antigen activated helper T cell and B cell move toward one another in response to check to chemokine signal and make the contact adjacent to the edge of primary follicles, CD40 ligands and CD40 ligand. So we see the antigen presentation to T cell activation, lymph nodes, the dendritic cells is it's activated and with the protein antigen, then interact with naive CD4 T cell, then the helper cells is activated, it expresses as normal, it expresses the CXCR5 and ligands and other things. Then CD4 ligands, it expresses CCX. This is if it has started to express this one, you see it has lost some of the characteristics expressing this one, and this one is lost some of the characteristics expressing something like CCR5, almost similar to the T cell when it is in normal state. So this is why they meet and other function happen. Another diagram is 12.8, antigen presentation on B cell to helper T cell. Protein antigen recognized by membrane immunoglobulin are endocytosed and processed, and peptide fragments are presented in associated with the class 2 major histocompatibility molecules. Helper T cell recognize MHC peptide complex and on the B cell and then stimulate B cell response. So in other hands, B cell receptor specific conformation after happen, you have the protein, there's B cell here, the B cell is standing as antigen presenting cells, then it will capture the peptide, it will be processed, then it will be expressed with MHC class 2, then with the MHC class 2 activated the CD4 already, will recognize that and it will stimulate the B cell to function, to differentiate and function further. So, in a T cell dependent B cell response to a specific protein antigen, at least two different epitopes of the proteins participate in process. A surface epitope on the native protein is recognized with high specificity by B cells, and a linear peptide epitope, which may be in any part of intact protein, is subsequently released by proteolysis bind to class II MHC molecules, and it is recognized by helper T cells. The antibodies that are eventually secreted are usually specific for conformational determinants of the native antigen, because membrane of Ig on the B cell is capable of binding a conformational epitope of protein, and the same Ig is secreted by a plasma cell derived from those B cells. This feature of B cells antigen recognition determines the fine specificity of the antibody response, and is independent of the fact that helper T cells recognize only linear epitope of processed peptide. In fact, a single B lymphocyte specificity for a native epitope may bind an endocytos, a protein, and present multiple different peptide complexes with class II MSC molecule to different helper T cells, but the resultant antibody response remains specific for native proteins. The principle outlined here for T cell B cell collaboration help to explain the phenomenon that is known as hapten carrier effect. Haptens such as dinitrophenol are small chemicals that can be recognized by a specific antibody but are not immunogenic by themselves. If, however, hapten are coupled to protein which serve as carrier, the conjugate are able to induce antibody response against the hapten. The hapten are equivalent to the conformational epitope that are recognized by B cell in T-dependent response to any protein antigen. Analysis of antibody response to the hapten carrier conjugate provide among the earliest demonstration of how antigen presentation by B cell lymphocyte contribute to the development of humoral immune responses. There are three important characteristics of hapten antibody response to hapten protein conjugates. First, such a response requires both hapten-specific B cell and proteins carrier-specific helper T cells. Second, to stimulate a response, the hapten and carrier portion have to be physically linked and cannot be administered separately. Third, interaction is class II MHC restricted, that is, the helper T cell cooperate only with B lymphocyte that expresses the class II MHC molecules that are identical to those that were involved in the initial activation of naive T cells by the dead T cell. All these features of antibody response to hapten proteins conjugates can be explained by antigen presenting function of B lymphocyte. Hapten-specific B cell bind to antigen through the hapten determinants, endocytose, and hapten carrier conjugate digest the protein component and present peptide derived from the carrier proteins into carrier-specific helper T lymphocytes. Thus, the two cooperating lymphocytes recognizing different epitopes of the same antigen. The hapten is responsible for efficient internalization of carrier protein into B cells, which explain why hapten and carrier must be physically linked. Because T cells recognize self-MHC associated peptide, the interaction with B cell is MHC restricted. Characteristics of humoral response is a response elucidated by hapten carrier conjugate applied to all protein antigen in which one intrinsic determinant, usually a native conformational determinant, is recognized by B cell and therefore analogs to the hapten, and another determinant is the form of class II MHC associated linear peptide is recognized by helper T cells and is an analog to peptide derived from the carrier protein. The hapten carrier effect is the basis of the development of conjugate vaccine against encapsulated bacteria. These vaccines contain carbohydrate epitopes recognized by B cells attached to proteins recognized by T cells, discussed later in this chapter. Role of CD40 ligands CD40 interaction in T-dependent B cell activation. Upon activation, helper T cell expresses CD40 ligands which engage its receptor CD40 on antigen-stimulated B cell and induce the B cell proliferation differentiation. Initially is extrafollicular fork and later in germinal center. CD40 is member of tumor necrosis factor receptor superfamily. Its ligand, CD40 ligands, which is CD154, is a trimetric membrane protein that is homologous to tumor necrosis factor CD40 in constitutively expressed on B cell, and CD40 ligand is expressed on the surface of helper T cell that have been recently activated by antigen and costimulators. When these activated helper T cell interact physically with antigen presenting cell B cell, the CD40 ligand to CD40 on B cell surface. This results in conformation alteration and preformed CD4 trimers, which induce the association of cytosolic protein called tumor necrosis factor receptor associated factor (TRAF) with a cytoplasmic domain of CD40. The tariff recruited to CD40 initiate enzyme cascade that lead to the activation and nuclear translocation of transcription factor, including the nuclear factor kappa beta and activating protein 1, which collectively stimulate B cell proliferation and increase the synthesis and secretion of Ig. Similar signaling pathways are activated by tumor necrosis factor receptors. CD40 ligand induced signal are also crucial for subsequent germinal center reaction, as well we will discuss later. In addition to T cell mediated dendritic cell and macrophage activated involves the interaction of CD40 ligands on activated helper T cell with the CD40 on this dendritic cell and macrophage. Mutation in CD40 ligands gene results in a disease called X-linked hyper IgM syndrome, which is characterized by the defect in antibody production, notably in isotope switching and family maturation, and deficiency in cell-mediated immunity. Similar abnormalities are seen in CD40 and CD40 ligase genes local twice. Interestingly, a DNA virus called the Epstein-Barr virus, Epstein ABV, infected human B cell and induces their proliferation. This may lead to the immortalization of the cell and development of lymphomas. The cytoplasmic tail of Epstein-Barr virus protein, latent membrane protein 1, associated with the same tariff molecule as does cytoplasmic domain of CD40, and this apparently triggers B cell proliferation. Thus, Epstein-Barr virus LMP1 is functionally homologous to the physiological B cell-like signaling molecule. The Epstein-Barr virus has apparently co-opted a normal pathway of B lymphocyte activation for its own purpose, which is to promote survival and proliferation of cell in which the virus can replicate. In addition to CD40 ligands on helper T cell activating B cell, helper T cell also secrete cytokine that contribute B cell response. T cell derived cytokine are essential for germinal center reactions described later. Several cytokines have been implicated with the early step of B cell proliferation and differentiation, and it is not clear if any are actually essential for this response. After the initial interaction of B cells with helper T cells at the interface between the follicles and T cell zones, subsequent activation of B cells by helper T cells can occur at two different locations, one outside the follicle in an extrafollicular focus and other in germinal center follicles. The nature of B cell response differ in this location. So we have activated helper, we have mechanism of helper T cell mediated B cell activation. Helper T cell that are activated by recognized antigen presented by B cell expresses CD40 ligands which bind to CD40 and on B cell and stimulate B cell proliferation and differentiation. Cytokine produced by helper T cell also contribute to B cell response. So we have activated helper T cell express CD40 ligands secrete cytokines. So we have CD40 lig. This is an B cell. They can meet with the T cell with the CD40 ligands. This lead to the B cell are activated by CD40 engagement cytokines. Then B cell proliferation differentiate. Extrafollicular B cell activation. B cell activation in the extrafollicular focus result in an early antibody response to protein antigen and set up subsequent germinal center reaction. Extrafollicular fork for T-dependent B cell activation generate low affinity antibody that can circulate and limit the spread of an infection. Each of such focus may produce 100 to 200 antibodies secreting plasma cells. In the spleen, extrafollicular fork develop in the outer portion of T cell rich periarterial and lymphoid cells, or between the T cell zone and the red pulp. And these collection of cells are also called pulse fork. Similar to T-dependent fork are observed in the medullary cord of lymph nodes. B cells that are activated by helper T cells through CD40 ligands in the extracellular fork undergo isotope switching. In fact, extracellular fork are the site at which most isotope switching occur during antibody response to protein antigen. The antibody secreting cells that are generated in the extrafollicular fork, including plasmablasts and tissue plasma cells, are mostly short-lived, and these cells do not acquire the ability to migrate to distant sites such as the bone marrow. In secondary lymphoid organs, plasmablasts they downregulate CXCR5 and CCR7, upregulate CXR4 and migrate to the red pulp in spleen or medullary cord in lymph nodes. In response to the CXR4 ligands, CXCL12, rather than taking a plasmablast properties, some of the B cell activated at the follicle T cell zone interface become precursor of germinal center B cell. FDC follicular dendritic cells release CXL13, which draw in small number, few to 100 of such cells. These precursor cells downregulate the EB12 and upregulate the receptor that promote their confinement to follicle center and initiate the germinal center reaction. The extrafollicular response also helps to generate T follicular help cells that migrate into the follicles and they are required for germinal center formation. T follicular help cells. Within 4 to 5 days after antigen exposure, activated antigen-specific B cells outside the follicular induce some previously activated T cell to differentiate into T follicular helper cell, which expresses high level of chemokines receptor CXCR5, are drawn into lymphoid follicle by CXL13. The ligand for CXR5 and play critical roles in germinal center formation and function. In addition to CXCR5, T follicular cells expresses ICOS, inducible co-stimulators, and programmed cell death protein 1, the cytokines interleukin 21, and transcription factor BCL6. The T follicular cells have phenotype that makes them distinct from T helper 1, T helper 2, T helper 17 subset of the effector T cell described in chapter 10. So differentiation of T follicular cells from naive CD4 T cell require two steps. Initial activation by antigen presenting presenting dendritic cell and subsequent activation by B cells. The choice between T helper 1, T helper 2, and T helper 17 fate on one hand, or the T follicular fates on the other depend partly on the strength of initial interaction between peptide class 2 MHC complex on dendritic cell and T cell receptor on naive CD4 T cells. A strong T cell receptor activation by dendritic cell induces T follicular cell by promoting expression of BCL6 transcription receptor and reducing the level of alpha chain of interleukin 2 receptor. This initial expression of BCL6 combined with weak interleukin 2 receptor signaling inhibit the acquisition of a T helper 1 to a 17 cell fate. Some of these activated T cells begin to express CXR, and their final differentiation into T follicular cell require interacting with activated B cells. A number of molecules on B cells and helper T cells are known to play a key role in generating T follicular cells. The binding of costimulatory molecule ICOS ligands and on activated B cell to its receptor ICOS a member of CD28 family on T cell promote the differentiation of the T cell into T follicular cells. T follicular cells generation also depend on the interaction between activated B cell and helper T cells mediated by a member of SLAM family. A signal molecule that associates with this SLAM family protein in T follicular cell is called SLAM-associated proteins, or SAP, signal stabilizing the expression of transcription regulator, particularly BCL6 that are required for T cell follicular development. SAP is mutated in patients with X-linked lymphoproliferative syndrome, which is associated with the defective in antibody and cytotoxic T cell response. So extracellular and the geminocenter B cell response. The features extracellular response, geminocenter response. So localization, extracellular follicular response occur in medulla cores of lymph nodes and at junction between T cell zones and red pulp spleen. Geminocenter is geminocenter of the secondary follicles. CD40 ligands is required for both extracellular response and geminocenter response. Specified T cells helps. There is extracellular extra follicular helper T cell, and there is also there is a T follicular cells in geminocenter. AD expression, yes to both. Isotope switching, yes to both. Somatic hypermutation is very low for extracellular helper T cell, but high in other parts. The terminal, in affinity maturation is low. Is low in extra follicular response than geminocenter. Terminal differentiated B cells, short-lived plasma cell in extracellular, in extrafollicular than in geminocenter. Transcription is BLIMP1 and transcription is BCL6. So sorry, figure 12:10, the generation of T follicular helper cells, follicular T cells. The generation of T follicular helper T cells require a sequential activation of T cells, first by dendritic cell and then by activated B cells. Inducible costimulator ICOS ligands, ICOS, this is the inducible costimulator ICOS ligands, ICOS L interaction are essential for T follicular cells differentiation, and the differentiated T follicular cells migrate into the geminocenter, where they activate B cells. Interleukins is B cell. Trying to eat something. So the defining cytokine produced by T follicular cell is interleukin 21. This cytokine is required for germinal center development and contribute to the generation of plasma cell in germinal center reaction. Interleukin 21 secreted by T follicular cells also facilitate germinal center B cell selection, event, and differentiation of activated B cell into plasmablast. In addition, interleukin 21 T follicular cells secrete other cytokines, including interleukin 4 and interleukin 13, and likely low level of interferon gamma as well. Some of these cytokines are known to participate in isotope switching, discussed later. Some extrafollicular T follicular cells may express cytokines such as interleukin 4 and interleukin 21, which stimulate isotope switching outside the germinal center as described shortly. The germinal center reaction. The characteristic event of helper T cell dependent antibody response, including affinity, maturation, generation of long-lived plasma cells and memory cells, and continuing isotope switching, occur in organized structure called germinal center that are created within lymphoid follicle during T-dependent immune response. The complex process of the B cell differentiation and selection of the cell with highest affinity antigen receptor that occur in this site is called germinal center reaction. Germinal center develop in secondary lymphoid organs approximately 4 to 7 days after initiation of T-dependent B cell response. Long before the functional significance of this anatomical structure was understood, it was named the germinal center by morphologists because the presence of many mitotic figure in the region suggesting that new cells are generated, germinated there. Germinal center consists of two distinct regions: dark zones, that is densely packed with rapidly proliferating B cells and appear dark in histologic section, staining with hematoxylin, and light zones where high affinity B cells are selected to survive, differentiate further, but more cell die that stain weak with the same dye. The cellular composition of these zones is different. T follicular cells are present only in the light zones. The light zones also contain types of a stroma cell called follicular dendritic cells. Follicular dendritic cells are not derived from the bone marrow precursor, and they are developmentally related to the fibroblastic reticular cells. They are found only in lymphoid follicles and express the complement receptor CR1, CR2, and CR3, and Fc receptor. These molecules are involved in displaying antigen for selection of germinal-center B cell, as described later. Follicular dendritic cells do not express class 2 MHC molecule, and in spite of their name, they are distinct from a class 2 MHC expression dendritic cell that present peptide to T lymphocyte. The long cytoplasmic process of follicular dendritic cell form a meshwork around which germinal center are formed. B cells in dark zones and light zones are sometimes called centroblasts and centrocytes, respectively. These zones are not fixed because B cells move between dark zones and light zones, and a rim of naive B cells in a follicular surrounding the germinal center makes up mental zones. The germinal center reaction consists of a number of sequential steps. First, initiation of germinal center by T follicular cells. The germinal center reaction begins with a migration of T follicular cells into a follicle guided by chemokine CXCL13 binding to CXCR5 on the T cells. Entry of B cells into the germinal center. Antigen stimulated B cells that have been activated in extrafollicular Fox cells through CD40 ligand interaction downregulate the AB12 and move into the follicular forms the germinal center. Each germinal center contains B cell derived from a little as well as few to up to about 100 antigen-specific clones. Different antigen-specific B cells clones may give rise to memory B cells and long-lived plasma cell. B cell proliferation. B cells that have been triggered by T follicular cells with the CD40 ligands CD40 interaction repeatedly proliferate, forming the dark zone for the germinal center. The doubling time of this proliferating germinal center B cell is estimated to be 60 to 12 hours, so that within 5 days, a single lymphocyte may give rise to as many as 5,000 progens. Somatic mutation in Ig genes. Proliferating dark zone B cell mutate their IgV genes as part of a process called somatic hypermutation. This process is initiated by the enzyme activating induced cysteine deaminase, which is highly expressed by dark zone B cell. The mechanism of somatic mutation is described later. B cell migration within a germinal center after multiple division. Dark zone B cell stop dividing and shut off expression of CXCR chemokine receptor. The CXCR4 ligand CXCR12 ligand is more abundant in dark zone than the light zone. The loss of CXCR4 expresses result in migration of these heavily mutated non-dividing B cells to adjacent light zone that contain T follicular cells and follicular dendritic cell. These non-dividing dark zone B cells still expresses CXR5 and are drawn toward the higher concentration of CXCL13 in light zone. Selection of high affinity B cell. In light zone, B cell can have undergone somatic hypermutation and are tested for their ability to recognize antigen displayed by FDC and free antigen in the germinal center. B cells that have acquired high affinity BCRs are more likely to capture antigen, display the antigen to T follicular cells and reactive CD40 and cytokine mediated signal from the T cell. As results, these B cells are selected to survive and differentiate further. B cell may fail selection and undergo apoptosis death in the light zone. This selection process is described later in more detail. Repetitive mutation and selection. B cells are positively selected in light zones expresses the transcription factor MEK re-express the CXCR4 chemokines receptor and thus return to dark zone. Germinal-center B cell can undergo repeated round of mutations and selection, migrating back to forth from light chain to dark zone if they are positively selected. This process contributes to the affinity maturation and antibody response. Differentiation into long-lived plasma cell. After some rounds of selection with affinity B cells exit the germinal center as plasmablasts that will home to bone marrow and differentiate into long-lived plasma cell. How the decision is made for selecting high affinity light zones B cell to not return to dark zone, but not to instead exit the germinal center is not known. What is known is that once this decision is made, high affinity light zone B cells re-expresses EB12 and leave the germinal center. Memory B cell formation. From the germinal center, B cells that undergo limited somatic permutation and remain relatively low affinity exit L as memory B cell. The memory cells have the ability to recirculate and migrate from one secondary lymphoid to another. That should be explained. So we see this is the germinal center. This is the expression of the germinal center in secondary lymphoid organ A. The germinal center is within the follicles and include basal dark zones, this dark zone, and adjacent light zones. Germinal center B cells expresses B cell lymphoma 6 as do in follicular helper cell. ICOS-inducible costimulatory expresses helper T cell reside mainly in the light zones and extracellular area. So germinal center formation is defective in human and in mice with genetic defect in T cell development and activation or with mutation of either CD40 or its ligand discussed earlier. Now that we have described the basic features and step of germinal center reaction, we will discuss some of the individual cellular and molecular events that occur during this process. Heavy chain isotope class switching. In T-dependent response, some of the progenies of activated IgM and IgD expressing B cells undergo heavy chain isotope class switching and produce antibodies with heavy chains of different classes such as Ig, gamma, alpha, and epsilon. Isotope switching outside the follicle is driven by extrafollicular pregerminal center T follicular cell, and the process continues to occur in the germinal center driven by T follicular cell in light zone. The capacity of B cell to produce different antibody isotopes provide a remarkable specialization of humoral immune response by generating antibodies that perform distinct effector function and are involved in defense against different types of infectious agents. B cells change the isotopes of antibody they produce by changing the constant region of heavy chain, but the specificity of antibody, which is determined by variable region, remain unaltered. The molecular mechanism responsible for the change in heavy chain constant region are described later. So actually this is the sequential event in the germinal center. Activated B cell migrate to follicles and proliferate, forming a dark zone on the germinal center. These B cells undergo somatic hypermutation, hypermutation of immunoglobulin Ig, VIV genes, and migrate to light zones where they encounter follicular dendritic cells displaying antigen and T follicular helper cells. B cells with the highest affinity Ig receptor are selected to survive and cycle back to the dark zone where they undergo somatic Hypermutation. Selected cells go back and forth multiple times and are repeatedly mutated and selected until they finally differentiate into antibody secreting cells. Memory B cells are derived from B cells that undergo through fewer rounds of somatic hypermutation and selection and emerge earlier from the germinal center than the antibody secreting cell. The antibody secreting cells reside in bone marrow and as long as lived in bone marrow as long-lived plasma cell, and memory B cells enter the recirculating lymphocyte pool. So actually, in our diagrammatic presentation is activation of B cell migration into germinal center. Then germinal center, you see there is dark zone here. There's B cell proliferation and somatic hypermutation happen. Then selection of high affinity because it will be going to follicular dendritic cell. So this follicular dendritic cell is like you'll be training them. The one with high affinity will be coming back, coming back, and then we will get a very with hypermutation we'll get a lot of good plasma cell, and plasma cell will go as memory cells. Plasma cell will go produce antibodies, and plasmablast can go to the bone marrow. So, isotope switching in response to different types of pathogen is regulated by a cytokine produced by helper T cells that are activated by this pathogen. Switching from IgM to IgG is prominent aspect of T-dependent antibody response against many bacteria and viruses. IgG antibodies do not only promote phagocytosis of opsonized microbes and activate complementary, but also transferred through the placenta to protect newborn, and they have longer half-lives in the blood than other isotopes. So the production of IgG contribute in many ways to the protective capacity of humoral immunity. In mice, switching to IgG subclass is induced by cytokine interferon gamma, which may be produced by T follicular cells activated by these microbes. However, there is no evidence that human isotope switching from IgG to IgG involve interferon gamma. Human T follicular cells that expresses high level of interleukin 21 relatively to interleukin 4 drive the IgM to IgG class switch, but the detail of which cytokine drives switching differently to IgG1, IgG2, IgG3, IgG4 remain to be ascertained. The humoral response to many helminth parasitic dominated by IgE antibody which participate in elimination of helminth. The IgE antibody also mediate intermediate hypersensitivity allergic reaction. Therefore, helminth likely influence T follicular cells, differentiate and induce these helper T cell to produce T helper cytokine interleukin 4, interleukin 3, which drive isotope switching from IgM to IgT. So, in summary, a humoral response to many helminth parasites is dominated by IgE antibody, which participate in elimination of helminth. IgE antibody also mediate intermediate hypersensitivity allergic reaction. Helminth likely influence T follicular cells differentiation and induce these helper T cell to produce T helper 2 type cytokines, interleukin 4, interleukin 13, which drive isotope switching from IgM to IgE. In addition, B cell in a different anatomical site switch to different isotopes in part because of the cytokines produced at this site. Specifically, B cells in the mucosal tissue which switch to IgA, which is antibody class that is most efficiently transported through epithelial into the mucosal secretion where they prevent microbes from entering through the epithelium. Switching to IgA is stimulated by transforming growth factor beta, which is produced by many cell types including helper T cells in mucosa and other tissue. Cytokine of tumor necrosis factor family, BAF and APRIL also stimulate switching to IgA because these cytokines are produced by myeloid cells that can stimulate IgA response in absence of T cell response or help. Some individual who inherit mutant version of TAC gene, which encode a receptor of this cytokine, have selective deficiency of IgA production. So here, immunoglobulin heavy chains, type switching, B cell activated by helper T cell signaling, undergo switching different immunoglobulin isotopes which mediate distinct effector function. Selected examples of switching are shown. All isotopes are capable of neutralizing microbes and toxin. Proliferation of inducing ligands is happening. So here we have IgM, this complementary activation. So if in a T follicular cell there is production of interleukin 21, interleukin 40, you are more likely to activate IgG, which IgG1, IgG3. When there is more expression of interleukin 4 and 13, IgE is possible. When this happens, activated mucosas happening cytokines like tumor growth factor, transforming growth factor, aprin, BAF, others, they can lead to the IgA production, which is mucosa immunity, transport IgA through epithelium. CD40 signal work together with cytokine to induce isotope switching. CD40 engagement induces expression of enzyme IDE, which we will see later. Is crucial for both isotope switching and affinity maturation. The requirement for CD40 signaling and IDE to promote isotope switching in B cell is well documented by analysis of mice and humans lacking CD40 ligands and IDE. In all these cases, antibody response to protein antigen is dominated by IgM antibody, and there is limited switching to other isotopes. Molecular mechanism of isotope switching is a process called switch recombination, in which the Ig heavy chain DNA in B cell is cut and recombined such that a previous formed VDJ exon that encodes the V domain is placed adjacent to downstream C region and intervening DNA deleted. The DNA recombination event involve nucleotide sequence called switch region, which are located in the introns between the J and C segment at this 5' and of each CH locus other than the delta gene. Switch region are 1 to 10 kilobases long, containing numerous tandem repeat of GC-rich DNA sequence, and are found upstream of heavy chain genes. Upstream of each switched region is a small 1 exon for initiator of transcription, preceded by 1 region promoter. Signal from cytokine-induced transcription from a particular 1 region promoter reading through 1 exon switch region and adjacent CH exon. These transcripts are known as germline transcripting because they are not translated into protein, but they are required for isotope switching to proceed. Germline transcripts are found at both mu locus and downstream heavy chain locus to which an activated B cell is being induced to switch. At each participating switch region, and germline transcript facilitates the generation of DNA double-strand breaks as described later. The DNA breaks is upstream mu switched region is joined to the break in the upstream selected switch region. As a result, the rearrangement of VDJ exons just upstream the mu switch region in the IgM producing B cell recombines with Ig heavy chain genes located in immediately after the transcription active downstream switch region. So this is the description. They say mechanism of heavy chain isotype switching. When antigen activated B cells encounter helper T cell signal, the CD40 ligands, and in this example interleukin 4, the B cell undergoes switching to immunoglobulin Ig isotope other than IgM in this example, IgE. This team initiate germline transcription through the I epsilon, S epsilon, the C epsilon locus, and proximal constant heavy genes are deleted, leading to recombination of VDJ exon upstream of the mu locus with the C epsilon gene. Switch region are indicated by cycle labeled SUS gamma S epsilon, I gamma mu, and gamma and I gamma, and I epsilon represent initiation site for germline transcription. So rearrangement of DNA in a germ producing cell, signal form helper T cells, then transcription through E locus, then a cleavage of recombinances, deletion of intervenes, then a recombinance happen, VDJ rings, transcription RNase, then translation Ig is produced. Cytokine determine which constant region will undergo germline transcription. For instance, interleukin-4 induced germline transcription through the I epsilon SSE locus. This leads to the production of germline E transcript in IgM expressing B cell, and then to recombination of the mu switch region with E switch region. The intervening DNA is lost and VDJ oxon is thus brought adjacent to E. The, the end result is the production of IgE with the same V domain as that of the original IgM produced by B cell. The key enzyme required for isotope switching and somatic hypermutation described later is AID. As we mentioned earlier, AID expression is induced in activated B cell, mainly the CD40 light signal from T follicular cells. The enzyme remove an amino acid group from the cytokines in a single-stranded DNA template, converting cytosine residue to deaminated uracils residue. How AIDS targeted to switch region is poorly understood. The enzyme has been propensity for certain geminal center containing tetranucleotide motif. Switch region are rich in these motifs, and cytokines induce the transcription through this region makes them accessible to AIDS. The relative specificity of AIDS for switch region compared with other regions of the genomes also containing similar motifs can be partially explained by the fact that these geminal center rich region contribute to the increasing stalling of the RNA polymerase II, which when stalled efficiently recruit aid switch region transcript tend to form at a stable DNA RNA hybrid involving the template strand DNA, thus freeing up the non-template strand, which form an open single-stranded DNA loop called the RR loop. The generation of single-stranded DNA by RR loop formations is critical because AID can target only single-stranded DNA. The RR loop is therefore a region where large number of C residue in switch DNA sequence are converted into new residue by AIDS. An enzyme called uracil N-glycosylates remove the U residue, leaving the abasic sites. An endonuclease called APE cleaves these abasic sites, generating a nick at each position, whereas RR loops facilitate the formation of discontinuum in the non-template strand of DNA, and break into double-stranded DNA require that nick also generated on the opposite template strand of DNA. The geminal center rich generated on the opposite template. The geminal center rich switch region RNA that remain tightly bound to a template strand DNA is degraded by protein complex called RNA exomes, thus exposing C residue transiently on the template strand and allowing A and U and G and AP to generate some nicks on this standard strand as well. Nick that are generated to both strands contribute to the double stranded break of both the S-mu donor switch region and in the downstream acceptors. Switch regions that is involved in the partial, particularly isotope switching event. The double strand breaks in the two switch region are joined together by use of a machinery involved in the double-strand DNA break repair and non-homologous end joining. In this process, the DNA between the two switch regions is deleted, and the net result is that original rearrangement of VJ chain is fused in the new constant region. So those highest description that we are seeing there here, the mechanism by which activation induced the deaminase generate the double-stranded breaks at two switch region. They say germline transcript form a DNA RNA hybrid in the switch region, and activation induced the deaminase the aminate C region to generate mu residue in single-stranded DNA. Uracil N glycosylase U and G remove the new residue to generate a base site where the apps endonuclease create nick that need to the double-stranded breaks, where these figures illustrate the only the generation of double-stranded breaks in the mu switch region. Similar double-stranded breaks occur around the same time in switch region for the downstream isotopes, thus facilitating switch recombination and isotope switching. So the amino center rich transcript allow the RR loop of the single-stranded DNA to form AID on the target single-stranded DNA. So this one has been target. Then you have AID convert C's to U's by deamination, get the C's to U's. Then uracil glycosylates create a basic sites, and then process continue. Affinity maturation, somatic mutation of immunoglobulin genes, and selection of high affinity B cell. Affinity maturation is the process that leads to the increased affinity of antibody for particular antigen as T-independent humoral response progresses, and it is the results of somatic mutation of Ig gene followed by selective survival of B cell that produce antibody with highest affinity. The process of affinity maturation generates antibody with increased ability to bind antigens and thus more efficient neutralize and eliminate microbes on their toxin. So see, this is the figure. There's figure here, figure 12.6. Helper T cells and 40 CD40 and CD40 ligands interaction are required for somatic mutation to be initiated, and as a result of affinity maturation is observed only in antibody response to T-dependent protein antigen. So let us continue. Sorry for a little bit delay. We see this is the somatic hypomutation, and we have been told that the helper T cell and CD40 and CD40 ligands are needed. Interaction are required for somatic mutation to be initiated, and as a result of affinity maturation is observed only in antibody response to T-dependent protein antigen. So in here we have VC. These are an overview of affinity maturation. Early in the immature response, low affinity antibodies are produced. During germinocenter reaction, somatic mutation of immunoglobulin Ig and the selection of B cells with high affinity antigen receptor resulting in the production of antibodies with high antibody for antigen. Ig proliferating germinocenter B cells in the dark zone rearrangement IgV genes undergo point mutation at an extremely high rate. This rate is estimated to be 1 in 10 power 3 V genes base pair per cell division, which is approximately 1,000 times higher than a spontaneous rate of mutation in other mammalian genes. For this reason, mutation in the rearrangement of Ig genes is also called somatic hypermutation. The V genes of expressed this heavy and light chain in each B cells contain a total of approximately 700 nucleotides. Thus, mutation will accumulate in expressed V gene region at an average rate of almost 1 per cell division. IgV gene mutation continued to occur in the progeny of the individual B cells. As a result, any B cell clone can accumulate more and more mutation during its life in germinocenter. It is estimated that as the consequence of somatic mutation, the nucleotide sequence of IgG antibody derived from one clone of B cell can diverge as much as 5% from original germline sequence. This usually translates into up to 10 amino acid substitution. The mutations are clustered in V regions, mostly in antigen binding complementary determining regions. And the presence of mutation correlates with increased affinity of the antibody for antigen that induce the response. The enzymes aid discussed earlier in the context of isotope switching also play an essential role in affinity maturation. They play essential role in the affinity maturation. The DNA deaminase activity of aid converts C residue to U residue that specify tetranucleotide IgCT hotspots that are found all over the genomes but are targeted primarily to rearrange the V region or in switch region as discussed above. Aid may recognize sequence in the location of rearranged VDJ exons, which explain at least partially why rearrangement of V region are highly suspected to the mutation. However, mechanism by which these rearrangement VDJ exons are specifically targeted is still unclear. The use of the US that are generated from CS may be changed to TS when DNA replication occur, thus generating a common type of C to T mutation, or the U may be excised by U and G. And the basic site thus generating is prepared by erraptron DNA repair process, eventually generating substitution with any of the 4 DNA nucleotides at each site of aid-induced cysteine deamination. Two enzymes, MSH2 and MSH6, involved normally in process of DNA mismatch repair, are important participants in the somatic hypermutation. MCH2 and MCH6 can recruit nucleoles that remove not only uridine and nucleotides, which are normally present in RNA, but also adjacent nucleotides. This mutated stretch is then repaired by error-prone DNA polymerase, thus extending mutation to a residue beyond the C residue that are targeted by AIDS. It's unclear how two well-known DNA repair mechanisms, base excision repair and mismatch repair, which are normally high-fidelity processes, recruit error-prone DNA polymerase in germinal-center B cell in the context of somatic hypermutation. Repeated stimulation of T cell dependent protein antigen in the light zone, as described later, lead to the migration of B cell into the dark zone and increased number of mutation in Ig genes of antigen-specific germinal center B cell. Some of these mutations are likely to be useful because they will generate high affinity antibodies. However, many of the mutations may result in the decline or even the loss of antigen bindings. Therefore, the next and crucial step in the process of affinity maturation is the selection of the most useful affinity B cells and type of Darwinian natural selection that ensures survival of the best B cells fittest the term of antigen findings, and this occur in the light zone. B cells that bind antigen in germinal center with high affinity are selected to survival. The L response to antigen result in the production of antibodies, some of which form the complex with residue antigens and may activate the complement. FDC expresses receptor for Fc portion of antibodies and for product complement activation including C3b and C3d. These receptors bind and display antigen that are complexed with antibodies and complement product. The antigen also may be displaced in three forms in a germinal center. Meanwhile, the germinal center B cell that have undergone somatic mutation migrate into a follicular dendritic cell rich right zones and germinal center. These B cells die by apoptosis unless they are rescued by recognition of antigen. Only B cells with high affinity receptor for the antigen are able to bind the antigen when it is present at low concentration. And these B cells survive preferentially because of several mechanisms. First, antigen recognition by itself induces expression of antigen apoptotic protein anti-apoptotic protein of BCL2 family. Second, high affinity B cell will preferentially endocytose and present the antigen and interact with the limiting number of T follicular helper cell in the germinal center. These T follicular cells may signal with the CD40 ligand and cytokine to promote survival of the B cell with which they interact. So that one and what can happen. So here with somatic mutation, immunoglobulin V genes, hybridomas were produced from spleen cell with mice immunized with 7 and 14 days previously with happen oxalosomes, coupled proteins, and from spleen cell obtained after secondary and tertiary immunization with the same antigen. So hydrodomas producing oxalazone-specific monoclonal antibody were produced, and nucleotide sequence of V genes encoding the immunoglobulin Ig heavy and light chain were determined. Mutation in V genes increase with time after immunization and with repeated immunization and is clustered in the complementary determining region. The location of CDRF3 in the heavy chain is approximate. The affinity of the antibody produced also tend to increase with the more mutation, as indicated by the low dissociation constant for hapten bindings. So we say as more antibodies produced, the more of antigen is eliminated and less is available in germinal center. Therefore, B cells that will be able to specifically bind to this antigen and can be rescued from death need to express antigen receptor with higher and higher affinity for the antigen. As a result, as antibody response to an antigen progresses, the B cells that are selected to survive germinal center produce Ig of increased affinity for antigen. This selection process results in affinity maturation of antibody response because somatic mutation also generates many B cells that do not express high affinity receptors for the antigen and cannot therefore be selected to survival. The germinal center are site for tremendous apoptotic death of B cell. DNA breaks associated with somatic hypermutation and isotypes switching predispose the chromosomal translocation of various oncogene into Ig genes lock, producing tumor of B cells, lymphomas. This explains why many lymphomas develop from germinal centers B cells. The germinal center may also contribute to the pathogenesis of autoimmunity if somatic mutation drive B cell clone in the germinal center to become strongly self-reactive. The response of the somatic mutated B cell to self-antigen is controlled by regulatory T cells, some of which expresses CX CR5 and are known as T follicular regulatory T cell. It is possible that deficiency of these regulatory T cells lead to the increased T follicular cells number and the survival of self-reactive B cell, resulting in autoimmune. B cell differentiation into antibodies secreting plasma cell. Plasma cells are morphologically distinct, terminally differentiated B cells committed to abundant antibody production. They are generated after the activation of B cells through signal from BCR, B cell receptor, CD4, TNTOREC receptor, other receptors including cytokine receptor. There are two types of plasma cells. Short-lived plasma cells are generated during T-independent response and early during the T-dependent response in extrafollicular cell in extrafollicular B cell fork described earlier. These cells are generated found in secondary lymphoid organ and in peripheral non-lymphoid tissue. Long-lived plasma cell are generated in T-dependent germinal centers in response to protein antigen, single from the B cell antigen receptor and interleukin 21 cooperate to generate the plasma cells and their precursor called plasmablast. Plasmablasts are the earliest cell in the lineage of antibody-secreting cells. They continue to proliferate like activated B cell, but express a little or no of CD20, the marker for mature B cell. Plasmablasts generated in the germinal center enter the circulation and home to the bone marrow, where they stop dividing and differentiate into long-lived plasma cells. These plasma cells are maintained by cytokines of BAF family that bind to the plasma cell membrane receptor called BCMA, which provide a signal that allows cells to survive for long periods. Typically, 2 to 3 weeks after immunization with T cell dependent protein antigen, the bone marrow becomes major site for antibody production. Plasma cells in the bone marrow may continue to secrete antibodies for decades after antigen is no longer present. These antibodies can provide immediate protection if the antigen is encountered later. It is estimated that almost half of the antibody in the blood of healthy adults is produced by long-lived plasma cell and is specific for antigen that were encountered in the past. Secreted antibody enter the circulation and because of secreting cells but mature plasma cell do not recirculate. So let's see the diagrammatic presentation. B cell selection in the germinocenter, somatic mutation of V genes, germinocenter B cells generate antibodies with different affinity for antigen. The binding of B cell to antigen displayed or on follicular dendritic cell is necessary to rescue the B cell from programmed cell death. B cell may also present antigen to germinocenter T follicular helper cell which promote B cell survival. The B cell with highest affinity for antigen thus have selective advantage for survival as the amount of available antigen decreases during an immune response. This leads to the average increase in affinity antibody for antigen as humoral immune response progress. So B cell is activated, naive B cell. There's antigen, this B cell activation by protein antigen helper T cell, migration into germinocenters. So B cell with the somatically mutated IgV genes, IgE with varying affinities. Then in here, B cell with high affinity antigen receptor bind to antigen on follicular dendritic cells, ingest antigen and present it, then they become more progressive, they can move up and they can continue. Those which are not able to do that, they will need to die. So the differentiation of B cells into antibody secreting plasma cell involve major structural alteration in the component of endoplasmic reticulum and circulatory pathway and increase the IgE production as well as change in IgE heavy chain from membrane to secretory form. The cell enlarge dramatically and the endoplasmic reticulum and Golgi apart become prominent. And the cell is transformed into a secretory cells that bear little or no resemblance to B lymphocytes. IgE production changes from membrane form, characteristic of B cell, to secretory form in a plasma cell because of change in carboxyl terminal of IgE heavy chains. For instance, in a membrane mu, C mu4 is followed by short spacer, 24 hydrophobic residue and cytosolic tail of 3 amino acid, lysine, valine, and lysine. IgM in secretory IgM, on the other hand, the C-mu domain is followed by tail pieces containing polar amino acid. This transition from membrane to secretory Ig is caused by alternative RNA processing of heavy chains messenger RNA. The primary RNA transcript in all IgM producing B cell containing the rearrangement VDJ cassette, the 4 C-mu exon coding for constant region domain and 2 exon encoding for transmembrane and cytoplasmic domain. Alternative processing of this transcript, which is regulated by RNA cleavage, alternative process which is transcript which is regulated by RNA cleavage and the choice of polyadenylation sites determine whether or not transmembrane and cytoplasmic exon are included in mature messenger RNA. If they are included, the mu chain produced contain the amino acid that make up the transmembrane and cytoplasmic segment, and it is therefore anchored in lipid bilayer of the plasma membrane. If on the other hand the transmembrane segment is excluded from the mu chain, the carboxyl terminus consists of approximately 20 amino acid constituting the tail piece. Because this protein does not have a stretch of hydrophobic amino acid or a positive charged cytoplasmic tail, it cannot remain anchored in the endoplasmic reticulum membrane and is secreted. Thus, each B cells can synthesize both membrane and secreted Ig. Most of the Ig heavy chains messenger RNA in the plasma cell is cleaved at the upstream polyadenylation site, so most of this messenger RNA is of secretory form. It is not known why differentiation of B cell to plasma cell is associated with the altered displacing of the IgRNA. All constant genes contain similar transmembrane exons, and heavy chains can potentially be expressed in membrane bound and secreted form. So this is the diagram of production of membrane and secretory immune chain in B lymphocyte. Alternative processing of primary RNA transcript result in the formation of messenger RNA for the membrane or secreted form of mu heavy chain. B cell differentiation results in increasing fraction of mu protein produced as secreted form. TP and TM and C refer to the tail pieces, transmembrane cytoplasmic segment, respectively. So you see the primary RNA transcripts, resting B cells. This is the B cell differentiation. It has taken here the membrane mu messenger RNA, transmembrane the cytoplasm, then this happened, the new amino acid are produced. Generation of memory B cells. Memory B cells are generated during the germinal-center reaction and are capable of making rapid response to subsequent introduction of antigen. Because memory cells develop mainly in the germinal center, they are primarily generated during T-dependent immune response. Although the majority of memory B cells develop in germinal center in a T-dependent manner, some IgM-expressing memory B cells are generated without T cell help and with little or no somatic hypermutation in a T-independent manner. Memory cells survive for a long period, apparently without continuing at antigenic stimulation, because they express high level of anti-apoptotic protein BCL2. Some memory B cell may remain in lymphoid organ where they were generated, whereas other exit germinal center and recirculate between blood and lymphoid organs. Most cells develop from memory cells develop from lower affinity antigen specific B cell that engage from germinal center relatively early after a limited round of somatic hypermutation and selection. The production of large quantity of isotype switching high affinity antibody is greatly accelerated after secondary exposure to antigen, and this can be attributed to the activation of memory cell. Many of the features of secondary antibody response to the protein antigens and their differences from primary response reflect the difference between responses of memory cells and naive B cells respectively. Effective vaccine against microbes and microbial toxin must induce both long-lived plasma cell producing high affinity antibodies and memory B cell. These will occur only in vaccine only if the vaccines are able to activate helper T cell. This concept have been applied to design of vaccine of some bacterial infection in which target antigen is a capsular polysaccharide which is incapable of stimulating T cell. In these cases, the polysaccharide is covalently linked to the foreign protein to form the equivalent of hapten carrier conjugate, which does activate helper T cell. Such vaccine, which are called conjugate vaccine, more readily induce high affinity antibodies and memory cell than do the polysaccharide vaccine without linked proteins. Conjugate vaccines have proved particularly effective at inducing protective immunity in infant and young children who are less able to make strong T-independent response polysaccharide than adults. So, properties of thermus dependent and thermus independent antigen. So thymus-dependent antigen, they are mostly proteins, while thymus-independent, polymeric antigen, especially polysaccharide, also glycolipid nuclei. When you speak of features of antibody response, so isotope switching for thymus-dependent is yes, and for thymus independence, low level, IgA to IgA. Affinity is yes, very high, but for thymus independent, no. Secondary response memory, yes, and for thymus, but no or less, only with C in polysaccharide. Role of transcriptional regulators in determining the fate of activated B cells. So the outcome of B cell differentiation is regulated by the induction and the activation of different transcription factor. It is clear from discussion so far that activated B cell can follow several fates. They can develop into short-lived, long-lived plasma cells which secrete large amount of antibody, or long-lived delayed memory cell which do not secrete antibody but survive for prolonged period and respond rapidly to antigen challenge. In chapter 10, we discussed the concept of that T cells fates are determined in large part by expression of various transcriptional activator and repressor. The same the same general principle applies in fate of activated B cell. The major transcription factor involved in determining the fate of germinocenter B cells are as following. BCL6. In the germinocenter, B cells signal derived through CD40 and interleukin 21 receptor induces the expression of BCL6, which function as the transcriptional repressor to maintain a germinocenter reaction, particularly the massive proliferation of germinocenter B cell. BCL6 repress the expression of cyclin-dependent kinase inhibitor and thus cooperate with transcriptional activators such as CMYB to accelerate rapidly cell cycle entry and germinocenter B cell. BSL6 also represses P53, a transcriptional factor that mediates cell cycle arrest and apoptotic death after DNA damage. As a result, dark zone B cell can tolerate the DNA break that accompany isotope switching and somatic hypermutation and are relatively resistant to apoptosis. BLC antagonizes another transcription repressor called the B lymphocyte-induced maturation protein, which is required for plasma cell development, thus prevents cell in germinocenter from prematurely differentiated into the plasma cell during germinocenter reaction. BLIMPs and ER4. BLIMPs, a transcription repressor, and ERF, the transcription activator, are induced in some of the activated B cell and commit these cells to plasma cell fate. In addition, suppressor BCL6 are the repressor that maintain the germinocenter B cell reaction. BLIMP suppresses a second transcriptional factor, PAX5, which is required for maintenance of mature B cell. Thus, BLIMP is permissive for plasma cell development. ERF4 continue to be expression of XBP1, a transcription factor that plays a critical role in unfolding protein response. XBP1 protect developing plasma cells from injuries as consequence of unfolding protein, which are produced as consequence of massive increase in protein synthesis, and contribute to the maturation of plasma cell and enhances synthesis of Ig seen in this cell. HHEX, relatively low affinity B cell that receive limited T cell help in light zone downregulate B cell fail to induce MYC have reduced the mTOR activation and differentiate into memory B cells. Memory B cells requires HHEX transcription factor that is induced in the absence of BCL6, and it it in turn induces the maintaining level of anti-apoptotic protein B cell 2, which is required for survival of memory B cells. Numerous other transcription factors are known to contribute to the germinal center B cell induction and maintenance. An important regulator of the germinal center B cell programs that represses other fates is batch. The transcription factor FOXO1 is required for development of dark zone germinal center B cell. It induces the expression of CXCR4, which is required for migration of the dark zone. In the light zone, PX3 kinase signaling downregulate FOX1. When B cells are positively selected in the light zone, they express MYC and FOX1 and migrate to the dark zone. Antibody response to T-independent antigen. Many non-protein antigens such as polysaccharide, lipids, and nucleic acid stimulate antibody production in the absence of helper T cell, and these antigen and the response they elicited are termed as thermos independence. These antibody response differ in several respects from the response to T cell dependent protein antigen. The antibodies that are produced in the absence of T cell helper are generally of low affinity and consist mainly IgM, with limited isotype switching to some IgG subtypes and also IgA. Subtypes of B cell that respond to independent antigens. The marginal zones and B cell subset of B cell are especially important for antibody response to Ti antigen, whereas response to T-dependent protein antigen are largely mediated by follicular B cell. Other B cell subset may be the primary responder to Ti antigens. Marginal zone cells are a distinct population of B cell that mainly responds to polysaccharides. After activation, these cells differentiate into short-lived plasma cells that produce mainly IgM. B1 cells represent another lineage of B cell that respond readily to T antigen, mainly in the peritoneum and mucosocytes. T-independent antibody response are initiated mainly in the spleen, peritoneal cavity, and mucosocyte. Macrophages located in the marginal zones surrounding lymphoid follicle in the spleen are particularly efficient at trapping polysaccharide when these antigens are injected intravenously. Ti antigen may persist for a prolonged period on the surface marginal zones or zone macrophage where they are recognized by specific B cells. Mechanism of T-independent antibody response. T-independent antigens are capable of stimulating B cell proliferation and differentiation in absence of T cells. The most important Ti antigen, polysaccharide glycoproteins, and nucleic acid cannot be processed and presented in associated with MSC molecules, and therefore they cannot be recognized by CD4 cell T helper cell. Most of Ti antigen are multivariant, being composed of a repeated and identical antigenic epitope. Such a multivalent antigen may cross-link many B cell receptor molecules on specific B cell, leading to activation without requirement of cognate T cell help. In addition, many polysaccharides activate the complement system by alternative or lectin pathway, generating C3d, which bind to the antigen and is recognized by CR2, thus augmenting B cell activation. As mentioned earlier, Ti response, which is another name when you say Ti, is T-independent. We say although Ti response typically exhibit little although Ti response typically exhibit little isotope switching, some Ti-independent non-protein antigen do induce Ig isotopes other than IgM. In humans, the dominant antibody class induced by pneumococcal capsular polysaccharide is IgG2. In mice engineered to back to the 40 ligands IgE, and many IgG subclasses are barely detected in the serum, but low level of IgE3 resemble human IgG2. And IgA is the serum in the serum were preserved. Cytokines produced by non-T cells may stimulate isotope switching in T-independent response, as described earlier in the absence of T cells. BAFF and APRO are produced by cells of myeloid origin, such as dendritic cell and macrophage, can induce the synthesis of AD in antigen-activated B cells through receptor of the BAFF receptor family called TAP. This may further facilitate the activation of T cell receptor on this B cell. In addition, cytokines such as transforming growth factor that help to mediate the IgA switch in the B cell are secreted by many non-lymphoid cells that at mucosal site and may contribute to the generation of IgA antibody mediated against non-protein antigen. Protection mediated by T-independent antibodies. The practical significance of Ti antigen is that many bacterial cell wall polysaccharides belong to this category, and humoral immunity is the major mechanism of host defense against infections by such encapsulated bacteria. For this reason, individuals with a congenital or acquired deficiency of humoral immunity are especially susceptible to life-threatening infections with encapsulated bacteria such as pneumococcus, meningococcus, and haemophilus. T-independent antibody also contribute to the generation of natural antibody, which are present in circulation for normal individual and are apparently produced without overt exposure to pathogen. Most natural antibodies are low affinity antibody, antibody, anti-carbohydrate antibodies postulated to be produced by peritoneal B1 cell. And responding to antigen from bacteria that colonize the gastrointestinal tract and by marginal zone B cell in the spleen. A large proportion of the natural antibodies in human and mice are specific for oxidized lipids such as lysophosphatidylcholine and phosphorycholine. These lipids are found on bacterial membrane and on the apoptotic cells, but they are not exposed on the surface of healthy host. Some experimental evidence indicate that natural antibodies specific for these phospholipids provide protection against bacterial infections and facilitates phagocytosis of apoptotic cell. The anti-ABO blood group antibodies, another example of natural antibodies, recognize certain glycolipid blood group antigen expressed on the surface of many cell types, including blood cells. Natural antibodies specific for blood group antigen are important barrier to blood transfusion and transplantation, but not important for host defense. Antibody feedback regulation of humoral immune response by Fc receptor. Secreted antibody inhibit continuing B cell activation by forming antigen-antibody complex that simultaneously bind to the antigen receptor and inhibit Fc receptor on antigen specific B cell. So here the feedback is very important. That's the mechanism of understanding even many other things. That antibody feedback regulation of humoral immune response by Fc receptor. A secreted antibody inhibits continuing B cell activation by forming antigen-antibody complex that simultaneously bind to antigen receptor and inhibit Fc gamma receptor on antigen-specific B cell. This explanation of phenomena called antibody feedback, which refer to the downregulation of antibody production by secreted IgG antibody. IgG antibody inhibit B cell activation by forming a complex with the antigen, and and this complex bind to the B cell receptor for the Fc portion of IgG called the Fc receptor 2, Fc RAB2, and the OCD22, CD32. So these are very important. The Fc gamma RA2B, or sometimes called the CD32. We will discuss Fc receptor in chapter 13. The cytoplasmic tail of Fc gamma RIIB contain an immunoreceptor tyrosine based inhibitor motif item. And items contain immunoreceptor based inhibitor motif items seen in chapter 7. When this Fc gamma receptor is engaged, the item on cytosol tail of receptor is phosphorylated and tyrosine residue, and it forms a docking site of phosphatase ship SH2 domain containing ionostatol phosphatase. The recruited ships hydrolyze a phosphatate and on the signal in a signaling intermediate phosphatidyl ionostatol triphosphate and inactivate this molecule. By this mechanism, engagement of Fc gamma RII2 RIIB terminate the B cell response to antigen. The antigen-antibody complex simultaneously interact with antigen receptor through the antigen and with Fc through the antibody. And this brings the inhibitory phosphatase close to antigen receptor whose signaling is blocked. So to explain this concept is BCR signal leading to PIP formation, which bind other signaling molecule leading to activation. This polyvalency you see. So normal Fc receptors, this is A, regulation B cell activation Fc gamma A, antigen antibody complex can simultaneously bind to the membrane immunoglobulin through antigen, and Fc receptor through the Fc portion of the antibody. So as consequence, this simultaneous ligation of receptor phosphatase associated with the cytoplasmic tail Fc inhibit signaling by B cell receptor. B cell complex block B cell activation. Items immunoreceptor tyrosine based inhibition. So Fc receptor mediated antibody feedback is a physiological control mechanism in humoral immune response because it is triggered by secreted antibody and block further antibody production. The importance of Fc RIIB mediated inhibition is demonstrated by uncontrolled antibody production seen in mice in which the gene encoding this receptor has been knocked out. A polymorphism in FcR gene have been linked to susceptibility to autoimmune disease, a systemic lupus erythematosus in humans. B cell expresses another inhibitory receptor called CD22, which is sialic acid binding lectin. Its natural ligand is not known, nor it's known exactly how it is engaged during B cell response. However, knockout mice lacking CD22 show greatly enhanced B cell activation, and the cytoplasmic tail of this molecule contain item tyrosine residue, which then phosphorylate the CRS family kinase, bind ST domain and tyrosine phosphatase SHP1. SHP1 remove phosphates from the tyrosine residue of several enzyme and adapter protein involved in B cell receptor signaling, thus abrogates the B cell activation. A mouse strain called more certain, which develops severe autoimmune with uncontrolled B cell activation autoantibody production, has naturally occurring mutation in the SHP1, conditional deletion of SHP1, as well as engineered loss of lin in B cell lead to the breakdown of peripheral B cell tolerance and development of autoimmune. Summary. In humoral immune response, B lymphocytes are activated by antigen and secrete antibody that act to eliminate antigens. Both proteins and non-protein antigen can stimulate antibody response. B cell response to protein antigen require contribution of CD4 T helper cell specific for antigen. T helper cell dependent B cell response to protein antigen require initial independent activation of naive T cell in the T cell zones and B cell in lymphoid follicles in the lymphoid organs. Each specific for different part of same protein antigen. A B cell that recognizes a conformational epitope of native protein antigen internalize the protein, process it, and display a peptide derived from the protein on its class 2 major histocompatibility complex or MHC molecule for recognition by helper T cell. The activated lymphocytes migrate toward one another and interact at the edge of follicles where the B cell present the peptide antigen to antigen specific helper T cell. Activated helper T cell expresses CD4 ligands which engage the CD40 on B cells, and T cells secrete cytokine that bind to cytokine receptor on B cell. The combination of CD4 and cytokine signals stimulate B cell proliferation and differentiation. Stimulation of activated B cell at extrafollicular site by helper T cell lead to the formation of extrafollicular fork where isotope switching occur and short-lived plasma cells are generated. Some activated helper T cell differentiate into specialized T follicular cell that express high level of inducible co-stimulator ICOS and CXCR5 and secrete interleukin 21. The T follicular cells and antigen activated B cell migrate into the follicles and T follicular cell activate these specific B cell to initiate the formation of germinal center. The late event in T cell dependent antibody response, including additional isotope switching, somatic mutation, affinity maturation, generation of memory B cell, and induction of long-lived plasma cell takes place within germinal center. Helper T cell derived signal, including CD40 ligands and cytokine, induces isotope switching in B cell by process of switch recombination leading to the production of various immunoglobulin Ig isotope. Isotope switching require the induction of activation-induced deaminase, a cysteine deaminase that convert cytosine to uracils in single-stranded DNA and different cytokine allow aid to access distinct downstream heavy chain lock. Affinity maturation occurs in germinal center and lead to the increased affinity of antibody during course of T cell dependent humoral response. Affinity maturation is the result of somatic mutation of Ig heavy and light chain genes induced by A, followed by a selective survival of B cells that produce high affinity antibodies and bind to antigen displayed by follicular dendritic cell in the germinal center. The high affinity B cells are best able to present antigen to T follicular cells, which promotes survival of B cells. Some of the progens of germinal center B cell differentiate into antibody secreting plasma cell that migrate to the bone marrow. Other progens become memory B cell that live for long periods, recirculate between the lymphoid organ and peripheral tissue and respond rapidly to subsequent exposure to antigens by differentiating into high affinity antibody secretors. The expression of various transcription factor controls the differentiation of activating B cell in plasma cell and memory cell. T-independent antigen are generally non-protein antigen that induce humoral immune response without involvement of helper T cells. Many T-independent antigen, including polysaccharide, membrane glycoproteins, and nucleic acid, are multivariant, can cross-link multiple membrane of Ig molecule on B cell and activate complement, thereby activate the B cell without T cell help. Toll-like receptor activation on B cell may by microbial product may facilitate T-independent B cell activation. T-independent antigens stimulate antibody response in which where is limited heavy chain class switching, affinity maturation, and memory B cell generation because these features are largely dependent on helper T cell, which are not activated by non-protein antigen. However, some T-independent isotope switching can be induced by toll-like receptor stimulation by microbes, which may lead to the production of cytokines of the tumor necrosis factor family that activate B cell to induce aid. Antibody feedback is the mechanism by which humoral immune response are down-regulated when enough antibody has been produced and soluble antigen complexes are present. B cell membrane Ig and the receptor on the B cell for Fc portion of IgG called Fc gamma RII2 are clustered together by antibody antigen complex. This activate an inhibitory signaling cascade through the cytoplasmic tail of SL that terminate the activation of B cell. There are more further readings that you can go through. The selected reading here. So we get prepared for the next chapter, which is effector mechanism of humoral immunity. As of now, have a nice time and enjoy.